The cell contains many structures inside, most importantly, the nucleus and the mitochondria, embedded within the cytoplasm, but what gives structure and shape to the cell is the cell membrane. The cell membrane also enables the cell to maintain a controlled intracellular environment, by selectively insulating the extracellular from the intracellular world, since a variety of substances must be transported into or out of the cell as needed, such as ions, water, and proteins. And the cell must also be responsive to environmental stimuli such as hormones and drugs. The cell membrane is composed of the three types of material present in all living cells, namely, lipids, carbohydrates, and proteins. Lipids are assembled in a lipid bilayer, constituting 50% of the cell membrane mass, and composed of phospholipids, cholesterol, and glycolipids. The phospholipids are the major type and are formed of a mixture of different compounds. Their fatty acids, Pha, have a polar head group and two hydrophobic tails. The heads of Pha contain phosphate and glycerol. They are hydrophilic, they love water, and are in close contact with the extracellular and intracellular environment. The tails contain saturated and unsaturated Pha. They are hydrophobic and situated in the core of the membrane. Unsaturated fatty acids have a greater mobility or fluidity within the membranes. This may directly affect how the proteins can move or function within the lipid bilayer. Cholesterol, on the other hand, tends to promote membrane rigidity rather than fluidity. Glycolipids contain carbohydrates in the form of oligosaccharides. They are found only on the extracellular face. Their function remains unclear, may control cell-cell interaction and may act as specific cell surface receptors. The hydrophobic tails in the core of the membrane associate to exclude water, and act as an effective insulator, preventing the passage of charged molecules, such as sodium, potassium and calcium. The lipid bilayer is not a free liquid, through which proteins float at random, but they have discrete structural domains, in which certain ion channels or enzymes are confined to specific areas, and are not free to diffuse around the cell. Transmembrane proteins are embedded in the lipid matrix. Transport of substances into or out of the cell, and transduction of transmembrane signals, are carried out by proteins, which are supported by the lipid bilayer, to keep them in the appropriate conformation. There is great diversity from cell to cell, among the types of proteins embedded in the plasma membrane. There are two classes of proteins, integral and peripheral. Integral membrane proteins do not dissociate from the plasma membrane, and may only be released through disruption of the membrane structure. While peripheral proteins penetrate the lipid bilayer, and can be dissociated from the membrane, without physically disrupting the membrane bilayer. The protein bundles can be utilized as receptors or hydrated ion channels. Receptors are sites for interaction with a hormone, a drug, or a ligand, an ion or molecule that binds to a central metal atom to form a complex. Hydrated ion channels are pores through which ions or other small molecules can pass into or out of the cell. This is a demonstration of sodium, calcium, chloride, and potassium ion channels. Sodium, calcium, and chloride channels promote inward currents by moving into the cell, while potassium channels promote outward currents by moving out. Channel proteins arrange themselves in a bundle to create a hydrophilic core filled with water, and a lipophilic, hydrophobic sides to interact with the membrane lipids. Receptors and channels are mainly formed of integral membrane proteins. These proteins can pass through the membrane, once or multiple times, to have their N-terminal and C-terminal, either on opposite sides, or on the same side of the lipid bilayer. An ion channel is a pore in the cell membrane, composed of proteins, assembled in subunits, alpha, beta, gamma, delta, etc. It allows selected ions to move passively, without the need for energy, in and out of the cell, depending on the concentration gradient. This is a demonstration of an ion channel, composed of proteins, assembled in subunits. Ion pumps, on the other hand, are energy dependent, and can move ions against their concentration gradients. Living cells require the orchestrated work of ion channels and pumps, to maintain appropriate ionic gradients, otherwise cell dysfunction or even cell death will ensue. You may think of the ion channel as having outer and inner vestibules, with a selectivity filter in between, which is highly specific for a single type of ions, e.g. sodium, potassium, or calcium. The gating function is the ability of a channel to open and close according to the cell needs. This function is regulated by certain signals, mediated by stimuli, such as mechanical stretch, voltage changes, and ligand stimuli. Voltage-gated channels have voltage sensors, which respond to changes in voltages across the cell membrane. Ligand-gated channels respond to ligands, hormones, or drugs. They can be regulated via external neurotransmitters such as acetylcholine, epinephrine, or adenosine. Acetylcholine interacts either directly with the channel proteins, or through muscarinic receptors, which consequently activate specific guanine-binding proteins, G proteins. Epinephrine interacts through the adrenergic receptors, which also activate specific G proteins. Adenosine interacts through the A1 adenosine receptors, which likewise activate specific G proteins. In conclusion, neurotransmitters can regulate ligand-gated channels through two mechanisms: either direct intramembrane protein interactions, or stimulation of receptors situated at the channel surface, which consequently activate specific G proteins. G proteins, in turn, modulate channel activity either directly via intramembrane interactions, or indirectly via a second messenger system, effector, such as cyclic adenosine monophosphate, CAMP, or cyclic guanosine monophosphate, CGMP. In addition to these types of channels, there is a different type, the leakage channels, with random gating, depending on an intrinsic rate of switching between the open and closed states. To perform their actions, the channel proteins must have regulatory regions. For example, G protein binding sites, voltage sensors, and sites for modifications, such as phosphorylation. 3 states of channels are identified: closed channel, ready to open; open channel, and inactivated channel, which is the phase of termination of the current in the open channel, and rearrangement of the channel protein to the closed configuration, then the channel is ready to open again. In addition to ion selectivity, ion channels, as stated earlier, are also classified into inward current channels, which promote depolarization, for example, sodium and calcium channels, and outward current channels, which promote repolarization or hyperpolarization, for example, potassium channels. In the resting, polarized state, when a reference electrode is positioned over the cell surface and a recording electrode is introduced inside the cell, the inside is found to be negative relative to the cell surface. Depolarization, reversal of polarity, is achieved by an inward current, sodium or calcium current, making the inside relatively positive. Repolarization is achieved by an outward current, potassium current, to restore the resting polarized state. At this stage, it is reasonable to discuss various types of ion channels, in some detail. Potassium channels in the heart are activated, via a selective G protein by acetylcholine, acting through muscarinic receptors, and adenosine, acting through A1 adenosine receptors. Potassium channel activation creates an outward current, positive potassium ions go out, making the inside of the cell more negative, and creating a more negative resting membrane potential, away from the threshold potential. This situation will result in repolarization, and may progress further to hyperpolarization. In this way, firing of the action potential is delayed, and action potential duration is prolonged, reducing the pacemaker firing rate. This figure shows the more negative resting potential, shown in red, created by potassium channel activation. This more negative potential takes longer to reach the threshold potential. Apart from acetylcholine and adenosine potassium channels, There are several other types of potassium channels in the heart: ADP depletion dependent, sodium dependent, delayed rectifying, correcting the depolarized state, and calcium activated rectifying potassium channels. In extracardiac tissues such as neurons, the muscarinic acetylcholine receptors can inhibit potassium channels, acting through other selective G proteins, contrary to what occurs in the heart. Thus, you can realize that there is too much variability of the potassium channels. Calcium channels are regulated by G protein-coupled receptors. Their activation increases intracellular calcium concentration, leading to muscle contraction. Two types of calcium channels are identified in the heart: transient, T type, mainly present in the atria, regulating the initiation of the heartbeat, and long-lasting, L type, mostly present in the ventricles, contributing to excitation-contraction coupling. These types are also present in the skeletal muscles, with non-defined effects. Other extracardiac calcium channels include P-type, first discovered in cerebellar Purkinje cells, and N-type, mainly present in neurons. Calcium channels are activated by beta-adrenergic receptors in the heart, via the stimulatory G protein, GS. On the other hand, calcium channels are inhibited by M2 muscarinic receptors, A1 adenosine receptors, A-type natriuretic peptide, ANP, and protein kinase C. Sodium channels carry the sodium inward current, responsible for the rapid upstroke, phase 0, of the action potential in atrial and ventricular muscle cells. Transmembrane receptors. Most transmembrane receptors do not transport extracellular substances from outside to inside the cell, but initiate a signal. Thereby these substances can regulate the intracellular function. Many receptor classes are identified. G protein-coupled receptors is the largest group. They utilize G proteins as intermediary between the receptor and an intracellular effector system, such as adenyl cyclase enzyme. They bind agonists, ligands, such as adenosine, catecholamines, acetylcholine, prostaglandins, angiotensin, vasoactive intestinal protein, and histamine. Tyrosine kinase receptors, such as epidermal growth factor and insulin receptors. Ligand transport receptors, which are exceptional in that they transport the ligand itself into the cell, such as LDL receptors. And ion channels receptors are present on the surface of ion channels, such as acetylcholine nicotinic receptor. Receptor pocket. The receptor pocket is the equivalent of the aqueous pore present in ion channels. It performs two functions: 1, it acts as the ligand binding site on the receptor surface, and 2, it determines the receptor selectivity for the ligands. Receptor G protein coupling. G proteins are complexes assembled in alpha, beta, and gamma subunits. More than 12 members of the alpha subunits are identified, the more common of which are stimulatory subunit alpha S, which activates adenylyl cyclase; inhibitory subunit alpha I, which inhibits adenylyl cyclase; subunit alpha 0, which is thought to regulate calcium channels; and subunit alpha Z, which is thought to activate phospholipase C. Alpha subunit structure. 3 sites are identified within the alpha subunit: receptor binding site, effector binding site, guanosine triphosphate, GTP, binding site. This site is responsible for the hydrolysis of GTP to GDP, guanosine diphosphate, mediated by the guanosine triphosphatase, GTPase, enzyme. The effector, adenylyl cyclase system. Adenylyl cyclase system consists of 1. The beta-adrenergic receptor, which recognizes the hormone epinephrine. 2. The stimulatory GS protein. 3. The adenylyl cyclase enzyme itself. Adenylyl cyclase can be stimulated, for example, by the beta-adrenergic receptor, which interacts with GS protein, or inhibited, for example, by the A1 adenosine receptor, which interacts with GI protein. Adrenergic receptor types and subtypes. Catecholamines exert their effects through their alpha and beta receptors. Alpha receptors could be subdivided into alpha 1 and alpha 2, and beta receptors are also subdivided into beta 1 and beta 2 subtypes. In addition, beta 3 subtype is identified predominantly in fat tissues, and at least 3 subtypes of each of the alpha 1 and alpha 2 subtypes were also identified. G protein subtypes. Likewise, a growing number of G protein subtypes were identified. Effector subtypes. Beta-adrenergic receptors do not only activate the effector system adenylyl cyclase, but can directly open calcium channels via GS, without first activating adenylyl cyclase. Similarly, the A1 adenosine receptor does not only act via adenylyl cyclase, but can directly open potassium channels. To further complicate matters, a single receptor subtype may be able to activate different G proteins, and even receptor signaling may be modulated by many conditions. Conditions modulating receptor signaling. Agonist-induced desensitization. Antagonist-induced sensitization. Hormones, particularly the steroid and thyroid hormones. Cyclic adenosine monophosphate changes. Clinical conditions, such as myocardial ischemia, heart failure, and diabetes mellitus. Agonist-induced desensitization is the attenuation of response with continued exposure to the agonist. Many mechanisms may be involved. Downregulation, inhibition of receptor synthesis with reduced receptor number, and/or receptor dysfunction, reduced ability to interact with G proteins, which could be due to uncoupling of the receptor from its G protein, or sequestration of the receptor away from its normal domain to a less accessible site. Thank you.